Good evening, everybody. Welcome to our OmniGenix call tonight. Excited to have you all joining us wherever you're calling in from. We want to welcome you. And I'm going to go ahead and launch into a screen share here so that we can spend a little time together talking about OmniGenix and learning what's captivating physicians worldwide. And if you're dialing in tonight and you've separated yourself from your family or left business a little bit early, thank you so much for that. We appreciate the time you're going to spend with us this evening. Well, we are right on top of the hour, and out of respect for everyone's schedule tonight, we're going to go ahead and get started. I'll make sure everything is set to go, and it is. All right, good evening, everyone. Welcome to our Regenerative Modeling for Exceptional Health Inside and Out webinar. Glad to have you joining us this evening. OmniGenix empowers your practice with the next generation breakthrough in regenerative medicine, and that is the topic tonight. And so we thank you for taking a little time out of your busy schedule tonight, and we're going to share some information. We're going to get through it pretty quickly, about 32 minutes, and we'll be out the door here. So want to start off talking about the FDA, and I want to be real clear here that the FDA currently does not approve any exosome products. But the good news is we have the only FDA-approved MSC, or mesenchymal master cell line, and we'll talk a little bit more about that as we go on through the evening. And our scientific breakthrough, we talk about it hitting new standards. What does that actually mean? Our target is empowering practitioners with the highest efficacy, systemically potent delivery of regenerative material in the market. I mean, really bringing something that's going to make a difference. And how do we do that? Exosomes is the topic for this evening, and as we step forward, really exosomes become our body's top influencer in corrective signaling. And we're going to get into the details about that. We have the only known exosome with validated pre-COVID single source in the marketplace, and that's a great standard to be a part of. The microRNA and RNA content has been validated. That's great news. We'll talk about that this evening. Perfect product replication based on cellular expansion. Now, that might not make any sense yet, but as we get into the details tonight, it certainly will. And camouflaged presentation for absolute cellular adoption. Proven components with validated COA on every vial. That means we're going to tell you exactly what's in it, and we're going to give you the confidence that every practitioner needs. Purity, potency, and consistency, those are the hallmark elements for OmniGenix. My name is John Altschuler. I'm the president of OmniGenix. We have with us tonight Mike Mooney and Cliff Layton. Mike Mooney is our OmniGenix lab partner. He's the VP of Business Development. And our discussion points tonight are really learning a little bit about our company, but also the what and the why about exosomes, application and opportunity for your practice, and some practitioner questions. We've had some questions here that are coming from doctors around the country, and talk a little bit about practice onboarding and a special offer for you and your practice this evening. Our mission, we believe, is pretty clear and pretty simple. At OmniGenix, we believe the most advanced science should be available to everyone, not just the elite. So our mission to take groundbreaking regenerative innovations and make them accessible, approachable, and effective for people to access them from you, their primary care provider, their specialty care provider. Our agenda tonight is really transformation and patient outcomes. Your practice can continue to be the trusted source that it is, and bringing in breakthrough wellness into your community is going to help you remain in that trusted position. The timing to engage this upgraded standard of care is unique in the market. And I think it is a very special time. We're now entering into really heightened conversations about regenerative medicine and particularly about exosomes. So with no further ado, I'd like to invite our guest to step forward. His name is Mike Mooney, and Mike has been in the space for quite a while, and I'm excited for you to get to hear from him because he has such an incredible wealth of knowledge and information and really has been a part of the emergence of the exosome story. And so, Mike, welcome. So glad that you are with us tonight. Maybe you could start off by giving us a little bit of your background.
Yeah, sounds great. So I've stumbled through and worked through the stem cell regenerative space for 10, 15 years now. I was lucky enough to be around, Chimera Labs, who I work with, obviously as one of their VPs, and helped launch their initial product years and years and years ago. And I've been lucky enough to see this industry grow, for better or for worse. Sometimes it's the Wild West, but it's starting to grow up a lot and quickly. And it's definitely clear that the FDA is working with the players that know what they're doing and are playing by the rules and submitting the right kind of data, and creating a foundation for the future. So yeah, I've been around this a long time. The exosome space can be really confusing because there's no real regulation around what you can call an exosome. So
Mhm.
finding the right team and the right science behind something that you can provide in your practice is absolutely important, and that's what we're here to talk about tonight.
Fantastic. Well, I know, and I think the Wild Wild West is actually a really great term for the space that we're in because people will say about anything, they'll the lack of validation or verification of the things that they say is pretty high in our industry. But tell us a little bit about the emergence of the exosome story. I know that you've been working with Dr. Duncan Ross for quite some time, and it's really an amazing story, and I'd love for you to tell it.
Yeah, so before Chimera Labs was exclusively an exosome manufacturer, we were the first to market many years ago, almost 11 years ago now. We were a stem cell research company, and that's what everyone at the time thought that was the future. And there's a lot of ways that you can use stem cells, but they have their limitations. So we were tracking our outcomes of efficacy, or I should say Dr. Ross was at the time, for things like COPD research. And eventually, once he started to play with exosomes to see what they were all about, the technology was barely there to actually dissect them and characterize them. And we had actually, as a company, had to create for years a way to actually characterize exosomes to see really what was going on with them, to understand them. But very quickly, the outcomes were being beat by the exosomes when we thought it was going to be the stem cells over and over again. So we had this aha moment where that was going to be the future. So we put our foot down and just focused on just that, and that's what we've been doing for over 11 years now. So that's turned into a major manufacturing process as a pharmaceutical company that's working with stem cells. We use the cells to create exosomes. We have a single donor source from 2018, so it's pre-COVID, no spike proteins. There's no vaccine or virus in our product. That's unique to us for an FDA-approved cell line. And it took a lot of money and a lot of time, about $30 million in 3 years, to convince the agency that this is how we were going to move forward into clinical trials. And that's exactly what we're doing. So most of our work now is we have our IND approval for stage 1, 2 clinical trials. We're on the phone constantly and having meetings with the FDA for stage 3 trials. So we're getting a lot of these started right now, and the future looks really bright.
Amazing, amazing. I know that Dr. Ross, I guess I would refer to him as the father of exosomes. I mean,
Yep.
he is the one that really has come out and made an enormous difference. And so I think there's a number of things that I really would love to hit on, and that is, the uniqueness of how the product is harvested. Where does it come from? And I think sourcing has become such a major, major question. Maybe you could talk a little bit about the sourcing. How unique is it? And really, is there anyone else in the market that is doing what you're doing?
It's a really good question. So sourcing is probably the most difficult thing for providers to understand because the reps are really obscure about it, or there's just so much out there, it's hard to navigate. At the end of the day, if you really want to understand your product, you have to be able to fully characterize it every single time you make a lot of product. So if it's not a placenta that's made into a master cell line, which by the way is more expensive than lyophilizing multi-donor approaches. So most products are multi-donor. There are a few cell lines that are out there. None of them are, have gone through the process with the FDA to get it characterized. They're just selling the product. But when you look at things like products that are from adipose or bone marrow or from cord blood, we're either using adult cells, which doesn't make any sense to me, or us, or better yet, if it's umbilical cord. Those are very similar MSCs, but there's not enough cells in that donor to create a master cell line. So you're going to have multiple donors. You're going to have a variance in your product every single time you make it. So each time we deliver a product, it's the same exact cell source, and it's going to continue to be because we're going to immortalize our cell line, which means we'll have the exact same cells forever. And with that, we can produce this almost like a drug, which is why the agency is favoring us so much compared to our competitors, who either A, aren't where we are yet, or aren't even into an IND status for clinical trials at all. So we're at the absolute front when it comes to clinical trial status with the FDA. We will probably have our first approval for BLA within, I would say, at quickest a year, but maybe 2. But no one's, I mean, maybe for a topical, approval, somebody's going to have that a little, as quick, but we're gearing up for not just topical applications, but full systemic IV applications, which is where our IND approval rating is at.
So there's a lot, I think there's, I don't know exactly how many producers there are of exosomes in the market, but there's a good number of them.
Mhm.
And they seem to get away with saying almost anything without having to provide any real data or evidence, or whether it's certificate of analysis or something that really speaks to the cargo. And so let's talk about that for a moment, because the potency, I mean, I think that we can basically say that the FDA with the approved master cell line has said the sterility is there, everything is acceptable for going into studies, something that becomes approved to be injectable as a research. But how does the components vary in terms of the cargo, which I think is a very big part of the discussion, because not everybody understands that, yes, there are growth factors and other things that are in that soup, so to speak, but the things that really deliver the significant payload is a very different topic.
Yeah, that's one of my favorite conversations. So over the course of 11 years, we've characterized in the exact same fashion that we characterize our process to a lot of our competitors, and we're just not seeing RNA being expressed by any of them, and negligible at best, with missing CD markers. So the thing is, it's so expensive to run that kind of operation. We've had about 100 million invested into our lab in the last 11 years. A lot of these exosome labs are really small. They use shared incubator space, or they're white labeled. So going through paying for all that characterization every single time is more about economics than it is about anything else. So for example, with the cord blood, if you were to do your third-party sterility, endotoxin, mycoplasma testing and fully characterize it, you'd have almost no product left, which is why having a placental master cell line is so important. It's just so much more expensive and more difficult to maintain. But to bring it home, we are crazy, because every single time we make a lot of product, our QC process is so dialed in that we go through all these, over a dozen different types of characterization. We do high performance liquid chromatography. We do DNA analysis on the cells before we produce. We do RNA sequencing on the back end. We know every protein and growth factor and their potencies for every lot of product that we make. That's insane to do that every time. The reason why we're doing that every time and spending all that money and those man hours, it takes us 8 weeks to make a lot. Most of our competitors are making product within maybe 2 to 7 days. The reason why we're doing that is for the future of our clinical trials. All of that counts as data. But the second thing is, every time we make a lot, we're making sure that those RNA bands are similar, consistent to the lot before that and the lot before that. If for some reason the ratios were off, that means something happened in our QC process, and we would have a product that wouldn't be consistent for our providers or our data. We would not send out that product to the public. We'd probably just use that for R&D. It hasn't really happened yet, but the point is, we're looking for that every time. What our competitors are doing is making a lot of product. They're doing their third-party testing, and then they do their particle count, which isn't actually a confirmation of an exosome. So the 100 billion or trillion count is just saying they have things of this size on a screen. They counted dots, but that could be microplastics or cell debris. It's not a confirmation of you having an exosome. So we actually know we have exosomes. We test CD markers. We test everything. We can actually take a photograph with one of the most expensive, microscopes in the world that we have, to actually look at these and use software to show the actual CD markers. We are the absolute only company in the world that does that.
Amazing. Well, I think that is one of the things that being able to clarify, to be able to show that difference is very, very significant. And I've heard Dr. Ross talk about the cargo is actually more important than the microvesicle count. And I think we get lost in this idea of how many billions or how many exosomes do we have, when really our focus should be what's the richness of the cargo and how is that able to be delivered?
Well, that's the whole point of an exosome. So when you talked about cell-to-cell communication, the way these cells communicate with each other, and all cells produce exosomes, we don't want all of them for regenerative, effect or clinical trials. But when you look at mesenchymal stem cells, the youngest ones possible ethically are placental. Those have the best data, the best instructions possible. So we have adult cells that are struggling, or that don't unlock, or they're not expressing genes that they used to express when we were younger, aging in a nutshell. And we can get cells to get back to unlocking those gene expressions that we need to get over serious things, serious indications and diseases, or even just anti-aging. You cannot do that without the RNA. If you take an exosome and it doesn't have an RNA function, or that RNA is not being expressed, it's technically no longer an exosome. You're taking the cargo that fell out of the bed of the truck, but it's missing the motor. So if you put it into the body, how does it know where to go if it doesn't use RNA to look for that inflammation, and deliver that microRNA to exchange with the cells. So the
Well,
whole idea is it's going to seek out inflammation. Almost every indication has an inflammatory response or process. It's going to go where it needs to go because that's the RNA.
so I get a couple pieces out of that. I get that this is like getting the building blocks on the site. So getting all the construction materials in one place is one aspect of it. And then having the architect show up that can tell everybody what to do is the other part of that. And then having all those things work in harmony. So
Mm-hmm.
I have a question coming in from a doctor in Scottsdale, and I'll ask this and their comment here is that the FDA has oversight, and my understanding is that exosomes or stem cells for that matter are not legal. How do we work with these products inside the regulatory environment?
So let's talk about legality. So they're not illegal, but they're not written as legal. So what the FDA can do, there's two things at play here at a federal level. They absolutely have authority over you as a provider on how you market indicated use or treatment claims that are not FDA approved. If
Hmm.
you have in any way any advertising or marketing around indicated use or treatment claims around stem cell products or exosome products, you can get a warning letter for marketing purposes. Those are not fun. No one wants to see anyone get those, but essentially you have to be very bleak and just vague on what you're doing with them. The FDA has no authority whatsoever on how you practice medicine with informed consent with your patients. So if your patient has a conversation with you about using something that's pre-approved, but in the sandbox of regenerative medicine, which is different from drug and device, it's an allowable area. It's just something that you have to market very carefully. But when you
Hmm.
speak to your patients and share data and success stories with other patients you've treated, that's all you need. Informed consent, you're an off-label approach. It's a little different with drugs, of course, but that's the whole story. It's as simple as that. Now
Perfect.
there is a great trend happening in Florida and Utah, and there's some other states gearing up for this, where they're actually making the states come out from the board position, passing legislation to make it fully legal. We hope to see more and more states adopt that.
Perfect. Okay, I have another question here from a doctor in Dallas. His question is, stem cells carry DNA and live cells, which can cause rejection or immune system related issues. How do
Mhm.
exosomes that are exogenous not present the same type of issues?
Well, the truth is a lot of the exosome products in the market are tissue products. And the way you can tell if you look at the vial, there's going to be a tissue ID number on them. We see a lot of products that are lyophilized or kind of blended that essentially they haven't purified the exosome. They're just in there. So they're exosome containing products. If there's tissue involved, they have to be a tissue lab. Some states require if your provider, like for example, I know New York, there's got to be more, but at least New York, if you carry stem cell products or products that contain tissue, you have to register as a tissue lab, which means you're going to get an FDA inspection every year or two minimum. That's a lot of work, and it's stressful and scary. You don't want those guys in your office for that reason. But the point is, tissue comes with a lot of complications and risk. Yes, there's DNA, there's antigens, there's cell debris, which is why you don't see a lot of systemic stem cell options in the United States. It's pretty much just joints in sports medicine, and because we can't culture expand cells within the guidance of the agency, you don't have enough cells to treat anyone systemically anyway with like Wharton's jelly or cord blood cells. It's just not really a systemic approach. Exosomes in our case at Chimera Labs, they are pure. So we have a few different products, but like Lux here, for example, our main product that's similar to our clinical trial product called Exoglow.
Hmm.
There's three ingredients. It is exosomes, it is saline and amino acids to help the process, but there's no tissue whatsoever.
Okay.
We're not even considered a tissue lab technically.
Okay. All right. I got two other questions for you coming in. One from a gal in San Diego. And she says, in my practice, I work with a lot of shoulders, knees, and spinal deterioration. Are exosomes able to deliver in these areas? Some cases, bone on bone or degenerative conditions. Also, should they be combined with stem cells or other growth factors?
So there's a mixture of methods that can work. I will say, no matter what you're using, if it's a bone on bone situation, you're swinging for the fence. If they're like, I should have had surgery 4 years ago, 5 years ago, you might be buying time with whatever the provider decides to use. I will
Hmm.
say that we've not conducted a joint study yet. We're going to work on that at some point, so I don't have any approval data to share. But I know providers out there are using this off label and having a lot of success with something that's not bone on bone. Again, that's anecdotal, but yeah, it's been going on for a long time. Now, cells, I think, have contention when it comes to conversation for what's good for a joint, but exosomes should be left out. Things like PRP, for example, that's a chondrotoxic, highly inflammatory thing we're still using that has maybe 30 growth factors, maybe 40, And if you're doing it on a 70-year-old that has osteoarthritis, well, those platelets are going to be not great, and that joint's not going to respond very well. So using something younger in theory is going to be your best option.
Mm-hmm. Yeah, I think the potency obviously coming from a mesenchymal cell of a placenta
Yeah.
versus somebody taking their adipose tissue, spinning it down, trying to get those growth factors, reinserting those, you're not going to get that type of benefit.
Yeah, treating aging patients with aging cells is never, in my opinion, the best approach. For what it's worth, mesenchymal stem cells, when they produce exosomes, those exosomes have hundreds of types of growth factors. So you have a lot more to work with. But again, the RNA is the most important part.
Okay, another doctor in Thousand Oaks, California. Have you seen improvement in neurodegenerative conditions or improvement in motor function for conditions like Parkinson's, ALS, things of that nature?
I mean, this is a joke I make all the time. We're the dumbest pharmaceutical company in the world because instead of creating one drug that has two indications like every other drug you've heard of, that's
Yeah.
two studies, we've created something that we have to do 300 different studies on, which is impossible to fund. So our future, yes, our future is absolutely, getting to the point of having indicated approval, insurance pathways for neurodegenerative indications, traumatic brain injury, stroke. One of our newest stage 2 clinical trials we're planning right now to get started really soon is renal failure. Even stage 5 patients we've taken off of dialysis with no transplant. So these are really powerful agents that have a huge future. But the idea is we got published in Nature a couple years ago for crossing the blood-brain barrier. Stem cells are way too large to do that, and when you do an infusion of stem cells, they get caught in the lungs. So these are 12 to 1,500 times smaller than stem cells, and they follow the inflammation. So in the case of anything neurodegenerative or TBI or stroke, theoretically, that's why they're crossing the barrier. Again, we know this happens, and if we can reduce inflammation in the brain and let that CNS reset, there's a lot of hope for a lot of these indications. Now, we won't necessarily cure something like ALS in the future. We don't know that's going to happen, but this is, but if you think about inflammation in any situation, if we were to reduce significantly the inflammation, especially in the brain, we would, we expect obviously some of those symptoms to improve.
Fantastic. Well, Mike, we're gonna invite you back because clearly we can't get through everything that we'd love to get through. So what final words might you leave with the practitioners who are looking at really taking advantage of what we might call the gold standard of our industry, or maybe jumping into regenerative medicine in a more significant way? What would your message be to them today?
All I can say is the last 2 or 3 years, I mean, the first 10 years almost of us doing this, it was just like us convincing the world that this is real. And then it's become, okay, this is trending. And now there's been this massive tipping point where we're holding on for dear life in such a good way because so many providers are seeing success and helping their patients, even if it's not on the insurance path. Take, for example, the wound care guys that have to go through all of these algorithms of care for getting certain patches approved. Well, what if they could maybe just have a patient skip the 1 or 2 years of pain for those non-healing wounds that are really non-compliant and say, okay, it's a little bit more expensive, but let's just get it done. And then those patients are telling their friends that it actually worked, and then their practice sees more new patients, and then there's more revenue, and there's this whole new model that comes with it. It's helping people in ways that are amazing. We've done, for example, the last 5 years in just the US alone, we've done over 500,000 cases with 0 adverse events. It's safe. It's just about having honest conversations about getting better healthcare. And frankly, since pandemic, patients are very commonly taking more and more intent into their own healthcare and paying out of pocket. And they're doing peptides and NAD and red light therapy, hyperbarics. So this is part of that conversation in functional anti-aging.
Mike, my hat's off to you. Thank you so much. I don't know if I'm going to be able to reparrot all the wonderful things that you've said, but we'll have you back, I promise.
Sounds great.
Thank you so much. So just a quick recap, everybody. I'm going to just run through a couple of data points that Mike really isolated quite well. I think that at the end of the day, patients want more than symptom relief. They want something that's going to transform and hopefully something that's going to last for many years to come. We understand that what we're talking about in terms of the source material is coming from the newborn. It's coming from human placental MSCs. And that's very important because all these things that are at their height in terms of their activity, that's the source material that we're utilizing. So whether it's energy or cellular communication or cardiovascular function or our innate immunity or hydration or our natural resilience or uncorrupted DNA expression, this is what we're trying to re-engineer as we move through the regenerative space. We understand that life happens and the influences on our body that we're constantly trying to keep up with, be it accidents, toxic exposure, the elements, gravity, gray hair, possibly wrinkles. But the bottom line is we need to be empowered with things that can help us overcome those a little bit quicker. Inflammation is certainly at the root of a lot of disease, if not all of disease, but that inflammatory signaling is very important to the process that we're using with exosomes. So as Mike mentioned, these nano-sized extracellular vesicles are 30 to 150 nanometers, which are very, very small. They do cross that blood-brain barrier very easily. Their function carry proteins, lipids, mRNA, microRNA to direct cell behavior, and that is the signaling part. And hopefully, what that leads to is repairing tissue, reducing that inflammation, and restoring function. So, the inflammatory signaling is really what's driving those mesenchymal cells to respond. And that can be summarized as homing into immune modulation, secretome release, tissue repair, and ultimately resolution. So these are biological conductors, powerful paracrine modulators. And so with the manufacturing process that we use, what can we be assured of? Well, we have the FDA approved master cell line, unmatched consistency because of how we process things. You heard Mike talk about the years in development, the investment that has been made, the amount of cases that we have had, and no adverse reactions. So what's important here is that our source material was actually created in 2018. That's before COVID. So it's pre-COVID, it's pre-vaccine for COVID, and absent of spike protein as well. Cultured in a xeno-free chemically defined media, isolated with multimodality assays for purity and potency so that you can see that. This is one of our clean rooms in our CGMP facility in Florida. This is the first page of a COA. So I was talking about the documentation that comes with the product. Here's the product where you have a matching lot number that is on that packing slip that refers to the COA. This is a standard that I think you'd be happy to have for your patients to review. So it's great information. We have that entire chain of custody. So some of the questions that are being asked by clients today is, I think that as we get more educated as the public, what's the source material? Where is the product coming from? And of course, Mike talked about some of these already tonight, but asking for that verified lot COA. I mean, it's really interesting what patients are asking for these days. How does the company identify an exosome? That just helps us evaluate our product in the market space. These CD markers that need to be there so that we know that what we're looking at isn't a little piece of microplastic that Mike mentioned, but it actually is an exosome. How much of that microRNA and mRNA is in the product? And then, of course, if it's lyophilized or they tell you it's shelf stable at room temperature, these are things that should raise an eyebrow. And we can dive into more of this anytime that you'd like to revisit with us. But the product assay reports, and we'll kind of finish up with this, is really just showing that we have done the analysis. We know what the protein concentration, we know what the exosome concentration is. These are a few of my favorite markers here on the screen showing the biological active units, showing the CD9, 63, and 81 that they're present. So it's complicated when you're comparing product in the marketplace, but we have all the information. Leave you with a couple of pictures though. This is a facial burn, someone pouring water on an oil fire, and not a great outcome. The good news is he knew Dr. Ross and he said, look, I got an experiment for you. Let's take these exosomes and spray them on your face twice a day. After 7 days, you start to see the tissue regenerate, or at least really smooth out. By day 60, you're looking at new skin, and not porous skin, but skin of like a baby. So it's quite impressive what happens topically and then what happens from the inside out. Here's microneedling 14 days apart. You can see a massive improvement in fine lines and wrinkles. We also have product that is great for hair restoration. If you're into that, then we have some treatment strategies. Best results are from injection. You can also microneedle this into the scalp. The follicle, going down about 1.8 to 3 millimeters, we're seeing some fantastic results there. So we all understand that as we age 50s, 60s, 70s, and beyond, we have other things that kind of creep in. It's amazing, and I think Mike stated this that it's difficult to have one product that can do so much because we have so many studies that we need to do. So there are hundreds of suppliers out there in the space. The question is, what are they missing? Well, they're missing a replicatable FDA-approved master cell line. They're missing a pre-COVID, pre-spike protein, pre-COVID vaccine, MSC donor. They're missing all the years of research and the investment. And they're, of course, missing the recognition of Dr. Ross, who is our innovator. If you want to learn a little bit more, you can certainly drop by our website on omnigenix.com. You can do that whenever you are ready. OmniGenix has a product line of really 3 different products. We have different strengths of each, but the Luxier is a predominant product that Mike was talking about tonight, between 1 and 15 trillion microvesicles per vial. Systemic application. Chiara is our secretome formula for topical, so scars, keloids, hair, and joints. And then we have Vibe, which is our topical for the estheticians, looking at microchanneling or microneedling. Very, very effective products. And so if we were to look at a practice scenario, if we were looking at the Luxer Plus product, our common use for us would be systemic application. Dosing would be dependent on condition, severity, and body weight. Our general suggestion is 1 to 3 of the Luxir plus 5 ml vials for first treatment. And looking at a 90-day treatment window, that would be a perfect time to get the product into a client's body and then wait to see where you are at the end of 90 days. So during that 90-day window, you might consider doing infusions every couple of weeks because our RNA content is most valuable for that first 2 weeks, and then it starts to dissipate. So these are all things that will come up with discussions with our team. The financial scenario is our vials. We have different sizes, averages around 500 ml or $500 an ml or less. The cash pay is 2 to 3 times. So when a client comes into your office, what are you able to charge them? That's really dependent on you and your practice, but there are excellent margins for practitioners, not to mention incredible treatment outcomes. So I'd like to introduce you to Cliff Layton. Cliff is the CEO of OmniGenix and has a great vision for what we're doing. I'd like to turn it over to you, Cliff. Thank you.
Yeah, thank you. And I feel like asking the audience, how would you like to follow these 2 guys? Great job. Hey, I want to thank everybody for taking the time, and I promise you we are a couple, 2 or 3 minutes from closing out this evening. History-wise, I got started in the regenerative medicine space really in the middle 1980s. Was very fortunate to be around and was part of actually creating a category which was called the super juice category, with a product that went on to set records that may never be touched. Through that experience, about the year 2000, started a company called Nature's Balance, and you probably have heard of that company as a supplement company. And so I've had a lot of experience with just how far you can get with a juice, with a nutraceutical, with natural products. And I'll be honest, no regrets, no regrets. Helped a lot of people in that time. 8 years ago, however, sold Nature's Balance, and then said, hey, I'm not quite done. Got involved with stem cells. And you know what? One of the things that Mike said this evening, and I hope you caught it because it's so true. We're at a time right now with exosomes that won't be repeated. There's an awakening, there's an epiphany that's happening to the mindset of consumers and practitioners, where they're beginning to realize that, wait a minute, there's something going on here with these exosomes. And Mike touched on it. And I've experienced it myself. For 8 years, we did work with people, thousands of people across the country with stem cells. We never did an injection without exosomes. And I'm thankful for the people that I was working with at the time that insisted upon that. And bear in mind, good exosomes, but not these exosomes. And yet, over the years, what we discovered was, from a true scientific standpoint, we were treating things that we know today you really can't treat with a stem cell. I don't want to, be the bearer of bad news, but we just found out that scientifically you really couldn't do it, and yet we were getting results. Now, I've done tons of double-blind studies in the nutraceutical space, and there's statistics that people that take a sugar tablet get results. Why? Because they're taking something they think works. So even if you take an account for that, over time you come to the realize that, wait a minute, could it be this little exosome? And that's exactly what we discovered. Mike made a point about that. I want to just sort of really make that point very, very clear to all of us on this call tonight, that they're real, they work, they do things that heretofore have not been done in medicine. We're not allowed currently to make some of the claims that we would like to make and can make, but like I said, the proof is in the pudding. As a result of that, one of the things we want to do this evening is we have a little bonus, sort of something that'll help some of the doctors that are here. And so for the next 72 hours, what we're going to do, and John's going to put it up on the screen here, it's very simple. If you place an order, and John's going to finish this up in a second, but if you place an order of $5,000 minimum, you'll get a $750 discount. So you'll pay $4,250 for that $5,000 purchase. If, on the other hand, you said, you know what, I actually need a little bit more, and you purchase $10,000 worth of product, we're going to give you a $2,000 discount. And I have to say, we don't have it on here, but if you're wanting a larger order than that, don't hesitate to talk to us, because this discount extends. And directions are on the screen here, but it's a very, very simple process to get set up. I'm going to sort of turn it back to John, let him take you through that. And once again, sincerely, I thank you for taking the time this evening to join us. And I look forward to speaking with you in person, hopefully in the very near future. Thank you.
Thank you, Cliff. I appreciate that. And for those of you, if you already have an account with us, you're already doing business with us, we're not going to leave you out. So you can take advantage of the same offer here as well. So I'm just going to give you a couple final steps here to get the discounted off of the orders that Cliff just mentioned. All you have to do is follow 4 simple steps. Visit our website, and that's omnigenix.com/intake. If you have not yet registered with the company, all that does is register you with the company. That will end up providing to you an email link, which you'll be able to activate your account. So all you have to do is click on that, go in and activate your account. Once you do, we will reach out to you with an onboarding call to just help you navigate that portal and make your first order. We'll help you do that. And then number 4, when ordering in the next 72 hours, you'll have a discount code. And here are the codes. OGEXO, so it's OmniGenix exosomes, like Exo, 750. That's if you go ahead and put an order in there, it's between 5,000 and 10,000, use that code. That's going to get you the $750 discount, $750 discount. If you have an invoice of over 10, it's going to go ahead and take off the 2,000 for OGEXO 2000, and you'll be able to get that essentially 20% discount. So let this special offer be the start of something amazing. We want to thank you for taking your time tonight to be with us, and we look forward to having you on our next call. And there are going to be some next calls. And so if you want to either get into our educational side of our platform and hear more information about treatment protocols and things of that nature, where we can talk a little bit more granularly about some of those things, get a little bit more education, figure out how this can fit into your practice. We certainly want to go there with you, and happy to do that at your request. So go ahead and reach out either to the OmniGenix team member that got you on this call tonight. They can certainly answer your questions, or you can plug into our system right here, and we'll come running. Thanks, everybody, for being with us tonight. Hope you have an awesome Tuesday. Thank you for being with us. Good night.